In 2012, the DNA of the 20th dynasty mummies of Ramesses III and another mummy believed to be Ramesses III's son Pentawa were analyzed by Albert Zink, Yahia Z. Gad and a team of researchers under Zahi Hawass, then Secretary-General of the Supreme Council of Antiquities, Egypt. Genetic kinship analyses revealed identical haplotypes in both mummies. Using the Witt-Athy haplogroup predictor, they identified the Y-chromosomal haplogroup E1b1a, EM2. In 2017, a team led by researchers from the University of Tübingen and the Max Planck Institute for the Science of Human History in Jena tested the maternal DNA mitochondrial of 90 mummies from Abu Zir el-Meleku in northern Egypt, near Cairo, which was the first reliable data using high-throughput DNA sequencing methods. Additionally, 3 of the mummies were also analyzed for Y-DNA. 2 were assigned to West Asian J and 1 to haplogroup E1b1b1, both common in North Africa. The researchers cautioned that the affinities of the examined ancient Egyptian specimens may not be representative of those of all ancient Egyptians since they were from a single archaeological site. Whilst not conclusive since the few relatively older mummies only go back to the 18th–19th dynasty, the rest being from then up to late Roman period. The authors of this study said the Abu Zir el-Meleku mummies closely resembled ancient and modern Near Eastern populations, especially those in the Levant. The genetics of the mummies remained remarkably consistent within this range even as different powers, including Nubians, Greeks, and Romans, conquered the empire. A wide range of mtDNA haplogroups were found including clades of J, U, H, HV, M, R0, R2, K, T, L, I, N, X, W. The authors of the study noted that the mummies at Abu Zir el-Meleku have 6 to 15% maternal sub-Saharan DNA, while modern Egyptians have a little more sub-Saharan ancestry, 15% to 20%, suggesting some degree of influx after the end of the empire. Other genetic studies show greater levels of sub-Saharan African ancestry in modern southern Egyptian populations and anticipate that mummies from southern Egypt would show greater levels of sub-Saharan African ancestry. Gordine, Anselin and Keita criticized the methodology of the Scheunemann et al. Study and argued that the sub-Saharan genetic affinities may be attributed to early settlers, and the relevant sub-Saharan genetic markers do not correspond with the geography of known trade routes. In 2022, Danielle Candelora noted several limitations with the 2017 Scheunemann et al. Study, such as its untested sampling methods, small sample size and problematic comparative data, which she argued had been misused to legitimize racist conceptions of ancient Egypt with scientific evidence. In 2023, Christopher Ehret criticized the conclusions of the 2017 study, which proposed the ancient Egyptians had a Levantine background based on insufficient sampling and a biased interpretation of the genetic data. Ehret argued this was reminiscent of earlier scholarship and also conflicted with existing archaeological, linguistic and biological anthropological evidence which determined the founding locales of ancient Egypt to be the descendants of longtime populations in northeastern Africa such as Nubia and the northern Horn of Africa. Because the 2017 study only sampled from a single site at Abu Zir el-Meleku, Scheunemann et al. 2022 carried out a follow-up study by collecting samples from 6 different excavation sites along the entire length of the Nile Valley, spanning 4,000 years of Egyptian history. 81 samples were collected from 17 mummies and 14 skeletal remains, and 18 high-quality mitochondrial genomes were reconstructed from 10 individuals. The authors argued that the analyzed mitochondrial genomes supported the results from the earlier study at Abu Zir el-Meleku. In 2018, the 4,000-year-old mummified head of Jehutinakht, a governor in the Middle Kingdom of the 11th or 12th dynasty, was analyzed for mitochondrial DNA. The sequence of the mummy most closely resembles a U5A lineage from sample JK 2903. A much more recent 2,000-year-old skeleton from the Abu Zir el-Meleku site in Egypt. Although no direct matches to the Jehutinakht sequence have been reported, haplogroup U5 is also found in modern-day Berbers from the Shiva Oasis in Egypt. A 2008 article by C. Cowdray, "The Complex and Diversified Mitochondrial Gene Pool of Berber Populations," recorded haplogroup U5 at 16.7% for the Shiva, whereas haplogroup U6 is more common in other Berber populations to the west of Egypt. In 2018, the mummified remains of two high-status Egyptian relatives, Nakht Ankh and Kachenam Nakht, were analyzed DNA by a team of researchers from the University of Manchester. The results found differences in the Y chromosome SNPs of the two mummies which indicated different paternal lineages, and concluded that Nakht Ankh and Kachenam Nakht were half-brothers, but Y chromosome sequences were not complete enough to determine paternal haplogroup. The SNP identities were consistent with mtDNA haplogroup M101 with 88.0591, 27% degree of confidence, thus confirming the African origins of the two individuals. A 2020 DNA study by Gad. Horace et al. Analyzed mitochondrial and Y chromosomal haplogroups from Tutankhamun's family members of the 18th dynasty, using comprehensive control procedures to ensure quality results. They found that the Y chromosome haplogroup of the family was R1b, which originated in Europe and which today makes up 50 to 90% of the genetic pool of modern Western Europeans. The mitochondrial haplogroup was K, which is most likely also part of a Near Eastern lineage. The profiles for Tutankhamun and Amenhotep III were incomplete, and the analysis produced differing probability figures despite having concordant allele results. Because the relationships of these two mummies with the KV55 mummy had previously been confirmed in an earlier study, the haplogroup prediction of both mummies could be derived from the full profile of the KV55 data. The 20th dynasty pair of Ramesses III and his son were found to have the haplogroup E1b1a, which has its highest frequencies in modern populations from West Africa and Central Africa, but which is rare among North Africans and nearly absent in East Africa. Quote, genetic analysis indicated the following haplogroups: Tutankhamun Y-DNA-R1b, mtDNA-K. Akhenaten Y-DNA-R1b, mtDNA-K. Amenhotep III Y-DNA-R1b, mtDNA-K. Yuya G2a, mtDNA-K. Tiya mtDNA-K. Thuya mtDNA-K. The specific clade of R1b was not determined. In 2010, Horace et al. Undertook detailed anthropological, radiological, and genetic studies as part of the King Tutankhamun family project. The objectives included attempting to determine familial relationships among 11 royal mummies of the New Kingdom. as well to research for pathological features including potential inherited disorders and infectious diseases. In 2012, Horace et al. Undertook an anthropological, forensic, radiological, and genetic study of the 20th dynasty mummies of Ramesses III and an unknown man which were found together. In 2022, S.O.Y. Keita analyzed 8 short tandem loci street data published as part of these studies by Horace et al. Using an algorithm that only has 3 choices: Eurasians, Sub-Saharan Africans, and East Asians. Using these 3 options, Keita concluded that the majority of the samples, which included Tutankhamun and Ramesses III, had a population affinity with Sub-Saharan Africans in one affinity analysis. However, Keita cautioned that this does not mean that the royal mummies lacked other affiliations, which he argued had been obscured in typological thinking. Keita further added that different data and algorithms might give different results, which reflects the complexity of biological heritage and the associated interpretation.